We have been discussing safety guidelines for demonstrations and for other laboratory activities, and I'd like to discuss some biological safety issues as well. The two principal ones that I want to discuss are dissection safety and sterilization and aseptic technique. There are also safety issues associated with biotechnology, with field activities, and so on. We have safety notes for all of these provided on the laboratory safety course website, and we encourage you to look at those and to review those as part of your safety training as well. So I just want to talk about two of them, and as I said, dissection safety and then sterilization. Dissection is an important part of biological lab activities, life science courses. Properly planned dissection activities are wonderful for really teaching students the principles of anatomy and physiology. What are some of the safety guidelines that will help you? First of all, the general guideline is to always provide a rationale for why you're doing a dissection activity. And it's best if this is provided in written form to address any concerns or issues that students or their parents might have. Secondly, preserved materials and preserved specimens, very important. The greatest concern, of course, is the origin of some of these preserved specimens. Always purchase preserved materials from reputable suppliers. They can guarantee that all of those specimens are obtained in accordance with all Department of Agriculture regulations for them. Almost all preserved specimens are initially preserved using formaldehyde. But then all of that formaldehyde is removed by rinsing. And so that even something that was initially preserved in formaldehyde is guaranteed to be 99.7% free of residual formaldehyde. The formaldehyde is necessary to fix the tissues, to cross-link the protein so no further tissue deterioration occurs. Rinsing procedures are usually specified with the specimens. Follow those directions carefully in order to preserve the life of those specimens. There is a certain degree of preservative odor that may linger, so we do recommend that all dissection activities be performed in a well-ventilated lab only. Some additional safety precautions for dissection procedures. Appropriate personal protective equipment, including gloves, apron, and safety glasses. Be sure that your dissection tools are quality instruments and that they are free of rust and so on. So you need to inspect all of those beforehand. Most of the accidents that occur with cuts during dissection activities are from either poor quality or old, rusty, dull scalpels and so on. You should rinse all formaldehyde specimens with water before dissecting. You can use a small amount of a formaldehyde-free preservative to keep the specimen moist. You don't want it drenched, but it shouldn't dry out as well because that's just going to make it harder to cut. You want to instruct students always on the proper technique for using scalpels and blades. You want to instruct them to cut away from their bodies. You want them to use scissors for the initial cutting of the rib cage and so on. That will be easier than trying to use a scalpel for that. Should properly mount the specimens. And finally, cleanup and disposal. It's very important when working with preserved materials that you wash your hands frequently and always at the end of the dissection activity. Students should be wearing gloves. You need to warn them about inadvertent contamination of parts of their body if they touch their face while they're wearing gloves, that they've been working with the preserved materials and so on. At the end of the activity, before leaving the laboratory, for proper chemical hygiene for any lab activity or biological lab activity, is to wash your hands. Cleanup and disposal, you want to make sure that you've planned enough time in the lab activity so that the students have time at the end to clean up properly, to wash their scalpels, to wash the dissection equipment, to wash the trays. If you're done with the dissection activity at that point, then the specimens should be double bagged for disposal, and you need to consult your local guidelines in terms of the proper disposal of that solid waste, what the guidelines are, what the regulations are, and what's allowed, and follow all of those biological waste disposal guidelines that are necessary. That talks a lot about dissection activity. How about sterilization and aseptic technique? This would be for microbiology type lab activities. These are popular labs. All students have had a throat culture at one time or another, so it's helpful to relate what that throat culture is to something that they would study in the laboratory. You want to be sure that you're working only with known, non-pathogenic microorganisms. Very important. Knowing that you're working with known, non-pathogenic microorganisms, you would then follow what are called the biosafety level 1 precautions. And these are actually printed by the federal government. It's called the Biosafety for Microbiological and Biomedical Laboratories Biosafety Level 1. And these would be considered standard microbiological practices, standard biological hygiene, if you will. Most important rule of standard biological technique is to wash your hands thoroughly with soap and water, frequently during the activity before handling any more microorganisms, certainly after removing the gloves, and then also at the conclusion of the microbiology lab activity. Biological hygiene is the same as chemical hygiene. No eating, no drinking, no smoking, no chewing gum, no handling contact lenses. You're wearing safety glasses as well. Don't apply cosmetics, don't store food there, anything else in laboratory work areas when you're working with microbiological organisms. There should be policies and procedures for the disposal of any sharps that might have been used. You want to perform all the procedures, being careful to minimize aerosols, because you don't want to create a fume or a dispersion of microorganisms. You want to decontaminate all work surfaces after the procedure. Anywhere that you have touched with or worked with the microorganisms, and the decontamination utilizes a 10% bleach solution. So you take a household bleach and dilute it 1 to 9, so tenfold. All cultures, stocks, and anything else that you've been used should be decontaminated prior to disposal. We'll talk about that sterilization technique. Always wear appropriate personal protective equipment, gloves, safety glasses, and so on. You want to observe your gloves, especially if you've been working with anything sharp. Make sure that there are no puncture holes and so on in the gloves. Sterilization procedures, before and you're working with it, you need to sterilize any of your glassware that you're using and so on. That can best be done with dry heat at 160 degrees. For practical purposes, you have really 4 techniques for sterilization: dry heat, filtration, chemicals, or autoclaving. The dry heat at 160 degrees for 2 hours, as we said, would be to use to sterilize any glass and metal lab equipment that you're going to be using as part of the microbiology lab. You can sterilize inoculating loops in a Bunsen burner flame. Materials that are contaminated with microorganisms have to be sterilized before disposal. Some of the materials that might require this sterilization include your disposable loops, petri dishes, biological culture media, culture tubes, and the gloves, actually, that were used to handle the organisms. There are two methods for sterilizing biological. One of them is autoclaving, and the other one is chemical sterilization. The autoclaving, you want to put that in a bag loosely tied, and at 30 minutes, 121 degrees and 15 psi pressure. Now, some states do have slightly different temperature and time requirements for biological waste sterilization, so you want to consult those. Chemical sterilization is to immerse anything in the 10% bleach solution that we described, and you want to immerse everything for at least 24 hours. Prior to disposal. I want to one last topic with respect to microbiology labs, and it's very popular to culture what we'll call household bacteria. You know, in other words, you might take a swab of the desk just to see what kinds of bacteria are normally growing, okay, or even from the human body and so on. You should never culture those household bacteria, for want of a better term, in an open dish, so a Petri dish. Really, because you would open, as long as you don't open it, never, then it would be all right. But anytime that's going to be open, you don't want to do that. And that's because when you culture it, you've taken something that is normally non-pathogenic, but now you've amplified it by a factor of about 100,000. And so you don't know what might happen there. And so even though they're known not to cause disease in the normal concentration, you don't want to amplify it. We recommend that if you're going to do anything with that type of surface bacteria and so on, that you use what's called Petri film, which provides a closed surface for the growth of the bacteria and is therefore safer. Anything that you work with, again, though, you have to perform the chemical sterilization at the end. We do have detailed instructions for all of these biological safety topics on this laboratory safety course website. Again, we have the dissection safety. I went through some of these fairly rapidly. I do urge you to consult those detailed instructions. If you have any questions about them, you can call us or email us, and we'll be happy to describe them. On the sterilization and aseptic technique, we actually do have drawings and instructions for how to for the aseptic technique, which I didn't describe here. So I urge you to consult that, and as always, if you have any questions, to get those questions answered before you perform the lab activities yourself or with your students. Thank you.